So now that we've addressed all of the metabolic pathways we'll look at in this course, I want to summarize briefly the regulation of each of those pathways in response to our 4 primary hormones. After doing this, we'll look at 2 specific examples where metabolic disturbances impact those pathways and you precipitate with some pathological symptoms. So the hormones we'll focus on are insulin, glucagon, epinephrine, and cortisol. And these 4 hormones should be very familiar to you at this point in time. In relation to insulin, insulin is high after a meal, and remember this is going to promote storage. It also promotes growth and protein synthesis. So the primary pathways we'll be impacting here are stimulating glucose storage as glycogen, fatty acid synthesis, and amino acid uptake. Glucagon, on the other hand, is elevated during the fasted state. So 3 to 4 hours after a meal, as blood glucose levels start to drop, we have an elevation of glucagon. Glucagon stimulates the mobilization of fuels as a means of maintaining blood glucose levels. So in order to do this, we're going to activate gluconeogenesis and glycogenolysis. It will also activate fatty acid release from the adipose as a means of supplying free fatty acids as a viable energy substrate for skeletal muscle and the liver. Epinephrine also stimulates fuel mobilization during stress. This will stimulate glycogenolysis in the skeletal muscle and, to a small extent, in the liver. And it will also simulate fatty acid release from the adipose. Cortisol, on the other hand, provides a more long-term requirement for maintenance of energy. Cortisol will be stimulating our amino acid mobilization from skeletal muscle, and will also stimulate gluconeogenesis as a means of enhancing transcription of the genes involved, and similarly it will stimulate fatty acid release from the adipose. So these will be our primary hormones we'll focus on, and now we'll focus specifically on the direct mechanism of action of each one of these during both the fed, fasted, and starvation states. It's critical that you know not just that insulin stimulates glycogen synthesis, but how insulin stimulates glycogen synthesis and what the regulatory enzymes are. And this is true for all of these pathways. Now let's review the pathways that are active in the fed state. Remember, in the fed state, insulin will be elevated and will be controlling the activity of these pathways. So glycogen synthesis will be active, as well as fatty acid synthesis and the synthesis of triacylglycerols. These will be our anabolic pathways. The catabolic pathways of glycogenolysis, gluconeogenesis, and beta oxidation will be inactive under the fed state conditions. The urea cycle will remain constant as a means of facilitating protein turnover in the body and excreting excessive protein from the diet. So let's briefly review the mechanism of insulin signaling. Insulin signals through a receptor tyrosine kinase. When insulin is bound, the receptor will autophosphorylate and recruit the IRS. Phosphorylation on the IRS will recruit GRUB2 by binding of the phosphotyrosine residues via the SH2 domain, and it will also recruit phospholipase C and protein kinase. The downstream action of this binding mechanism is multifaceted. The initial response of insulin-sensitive tissues is shown here, where upon insulin binding to the receptor, this initiates translocation of GLUT transporters to the plasma membrane. These GLUT transporters are primarily GLUT4, and this activity is prevalent on skeletal muscle and adipose. GLUT4 is an insulin-sensitive GLUT transporter, meaning its recruitment to the plasma membrane is dependent on insulin being present. You can review the different types of GLUT transporters in Table 27.5. GLUT1, which is insulin insensitive, is primarily on the red blood cell. And we know that red blood cells are reliant on glucose as an energy source as they lack mitochondria. GLUT2 is insulin insensitive and is primarily found on the liver. And again, GLUT4 is our insulin-sensitive transporter that is recruited to the plasma membrane in response to insulin and is primarily found on adipose and skeletal muscle. Once insulin is bound, it will regulate several different pathways, the first of which being glycolysis. Once glucose has entered the cell, it's rapidly phosphorylated to glucose 6-phosphate. And this is facilitated by glucokinase or hexokinase, depending on the tissue you're in. Remember, glucokinase is primarily in the liver and requires a high level of glucose in order to be active, whereas hexokinase is in peripheral tissues and will much more rapidly phosphorylate glucose. So once glucose is phosphorylated to glucose 6-phosphate, there are two other primary regulatory steps in glycolysis. The first being phosphofructokinase 1, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. This is activated by AMP as well as fructose 2,6-bisphosphate. Our second regulatory step is the conversion of phosphoenolpyruvate to pyruvate. The enzyme here is pyruvate kinase, and this is stimulated by fructose 1,6-phosphate here, and can be inhibited by phosphorylation or by alanine. These are our three regulatory steps in glycolysis. The regulation by fructose 2,6-bisphosphate is again shown here. In the case of high levels of glucose, such as in the fed state, fructose 6-phosphate can be converted to fructose 2,6-bisphosphate by the enzyme phosphofructokinase 2. This is a bifunctional enzyme, and in the dephosphorylated state, its kinase activity is active. Fructose 2,6-bisphosphate in turn stimulates phosphofructokinase 1 and increases the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The next pathway that's primarily active in the fed state is glycogen synthesis. The regulation of glycogen synthesis is shown here, where when insulin is high, a phosphatase will dephosphorylate glycogen synthase, and glycogen synthase will be active in the dephosphorylated state. Under these conditions, glycogen synthase will accept UDP glucose and use it as a substrate to synthesize glycogen. This synthetic process can occur in both the liver and the skeletal muscle. Our other synthetic pathway that's also occurring is fatty acid and triacylglycerol synthesis. This is summarized here, where under high levels of glucose, in addition to glucose being converted to glucose 6-phosphate and being used to synthesize pyruvate, glucose 6-phosphate can also be accepted by glucose 6-phosphate dehydrogenase and used in the pentose pathway. The pentose pathway generates NADPH, which is required for fatty acid synthesis. Fatty acid synthesis also requires citrate, which under high levels of glucose, pyruvate will be converted to acetyl-CoA, and acetyl-CoA will condense with oxaloacetate, and this citrate, when it becomes elevated, will be shuttled out of the mitochondria and used as a substrate to synthesize malonyl-CoA. Malonyl-CoA will be our required substrate for fatty acid synthase to generate palmitate. The summary of this regulation is shown here, where citrate will stimulate the activity of acetyl-CoA carboxylase, which is our regulatory enzyme for fatty acid synthesis. Insulin again will also stimulate a phosphatase, which will dephosphorylate acetyl-CoA carboxylase, converting it to the active state, such that acetyl-CoA carboxylase can facilitate the synthesis of palmitate. Palmitate is a fatty acid that will be synthesized into a triacylglycerol and packaged as a VLDL within the liver. The VLDL will carry the triacylglycerol through the bloodstream to the adipose, where it will interact with lipoprotein lipase through the activity of ApoC2 and will free fatty acids, which can be stored in the adipose. Remember that storage is in the form of triacylglycerol. Consequently, these free fatty acids will recombine with a glycerol 3-phosphate to synthesize the triacylglycerol. Those are our primary metabolic pathways in the fed state. Keep in mind, in the fed state, the primary fuel for most tissues is glucose. Glucose is taken up by the peripheral tissues using these GLUT transporters. In the liver, it's insulin-insensitive GLUT2. In skeletal muscle and adipose, it's GLUT4, which is insulin-sensitive. And in the brain and red blood cells, it's primarily GLUT1. Now let's transition to the fasting state. Under fasting conditions, glucagon is elevated. Cortisol and epinephrine may also be elevated under these conditions. Activation of these hormones stimulates glycogenolysis, gluconeogenesis, lipolysis, beta oxidation, muscle catabolism, and ketone body synthesis. The urea cycle is also elevated under these conditions, as deamination of amino acids required for gluconeogenesis is necessary, and the amine groups from those amino acids must be converted into urea. So the initial activity of glucagon is shown here. Glucagon binds a G protein coupled receptor. Epinephrine can also bind a G protein coupled receptor. In both cases, this activates adenylate cyclase, and cyclic AMP is our second messenger for this cascade. Cyclic AMP activates protein kinase A, which is responsible for phosphorylating and activating our enzymes that are required for the fasted state metabolism. In the case of glycogenolysis, protein kinase A phosphorylates phosphorylase kinase, activating it, and phosphorylase kinase phosphorylates glycogen phosphorylase. Glycogen phosphorylase facilitates the conversion of glycogen to glucose 1-phosphate, which is converted to glucose 6-phosphate until it's dephosphorylated by glucose 6-phosphatase and can help facilitate the maintenance of blood glucose. This is the mechanism by which glucagon or epinephrine can regulate glycogenolysis in the liver. Keep in mind the mechanism by which epinephrine regulates glycogenolysis in the skeletal muscle is slightly different, and you should feel free to revisit that. The other pathway that's active and required for maintenance of blood glucose is gluconeogenesis. Gluconeogenesis primarily occurs again in the liver and is an additional source of glucose. Gluconeogenesis is essentially a reverse of glycolysis, except for the 3 regulatory steps, which must be bypassed in glycolysis and are alternative enzymes in gluconeogenesis. First off, glucagon via protein kinase A will phosphorylate and inactivate pyruvate kinase. This allows for the conversion of pyruvate to oxaloacetate in the mitochondria by pyruvate carboxylase. Remember, pyruvate carboxylase is allosterically activated by acetyl-CoA, and the acetyl-CoA under these conditions is coming from beta oxidation. Oxaloacetate can be shuttled out of the mitochondria as malate and can be converted to phosphoenol pyruvate using the enzyme phosphoenol pyruvate carboxykinase. The coupled reactions of pyruvate carboxylase and phosphoenol pyruvate carboxykinase bypass the enzymatic reaction of pyruvate kinase. From here, phosphoenol pyruvate will follow the reversible steps of glycolysis up to fructose 1,6 bisphosphate. From here, fructose 1,6 bisphosphate is converted to fructose 6-phosphate using the enzyme fructose 1,6 bisphosphatase. This will bypass our step regulated by phosphofructokinase 1 in glycolysis. This reaction is elaborated on a little more here, where we can see fructose 1,6 bisphosphate being converted to fructose 6-phosphate by the enzyme fructose 1,6 bisphosphatase. In the presence of glucagon, cyclic AMP will activate protein kinase A, and protein kinase A will phosphorylate the enzyme fructose 2,6 bisphosphatase. This will activate the bisphosphatase and convert fructose 2,6 bisphosphate into fructose 6-phosphate. The decrease in fructose 2,6 bisphosphate reduces the inhibition of the enzyme fructose 1,6 bisphosphatase and will facilitate the conversion of fructose 1,6 bisphosphate to fructose 6-phosphate. Keep in mind that the enzymes phosphofructokinase 2 and fructose 2,6 bisphosphatase are a single bifunctional enzyme, and the activity of a phosphatase or a kinase is dictated by whether the enzyme is phosphorylated or not. So in the fasted state, the enzyme is phosphorylated and the enzyme functions as a phosphatase to decrease levels of fructose 2,6 bisphosphate. In the fed state, the enzyme is dephosphorylated and referred to as phosphofructokinase 2, and the enzyme functions as a kinase and phosphorylates fructose 6-phosphate to fructose 2,6 bisphosphate. The other pathways that are active are both lipolysis and beta oxidation. This is shown here, where under low levels of insulin, or high glucagon, and also epinephrine, cyclic AMP activates protein kinase A, which will phosphorylate hormone-sensitive lipase. Hormone-sensitive lipase in the adipose will hydrolyze triacylglycerols into free fatty acids and glycerol. These free fatty acids can be used as an energy substrate by both the liver and skeletal muscle, and oxidized to acetyl-CoA. Glycerol can be used as a substrate for gluconeogenesis. Remember, the acetyl-CoA generated by fatty acid beta oxidation is not a viable substrate for gluconeogenesis. Additionally, under these conditions, glucocorticoids or cortisol also facilitates lipolysis, and, more importantly, it facilitates the mobilization of amino acids from skeletal muscle. These amino acids, primarily alanine and glutamine, are our primary source of carbons to synthesize glucose in gluconeogenesis. You can see here that cortisol facilitates this process, as well as, again, lipolysis, gluconeogenesis, and glycogenolysis, all as a means of supplying blood glucose. The interaction of these pathways under fasted states conditions is summarized here, where again, lipolysis from the adipose supplies free fatty acids and glycerol. The glycerol can be used as a substrate for gluconeogenesis to sustain blood glucose, and the free fatty acids can be used as an alternative energy source by both the skeletal muscle and the liver, where they're beta oxidized to generate acetyl-CoA. Acetyl-CoA in the skeletal muscle is oxidized by the TCA cycle to generate ATP, and acetyl-CoA in the liver is also oxidized by the TCA cycle, but can be used as a substrate to synthesize ketone bodies. Under the fasted state conditions, ketone bodies can be used as a substrate for energy by the skeletal muscle. The skeletal muscle also provides amino acids for gluconeogenesis in the liver as a means of, again, synthesizing glucose to be used to maintain blood glucose levels. The deamination of these amino acids provides ammonia that is used by the urea cycle to synthesize urea, and urea is excreted in the kidney. The brain and the red blood cell use glucose as a primary fuel substrate, and the red blood cell synthesizes lactate under anaerobic glycolysis as it lacks mitochondria, and the lactate can also be used as a substrate for gluconeogenesis to synthesize glucose. So this readily summarizes the interaction of these pathways under the fasted state conditions. And keep in mind, the regulation of all of these pathways is also essential. The fuel usage in the fasted state is summarized here, where again the brain and the red blood cell are again using glucose. GLUT1 is an insulin insensitive transporter. The adipose, the skeletal muscle, and the liver are primarily using fatty acids, although the skeletal muscle can utilize any ketones that are synthesized. And under these conditions, skeletal muscle and adipose are not using any glucose, as their glucose transporters are insulin sensitive and insulin is low in the fasted state. The phosphorylation states and the activity of the regulatory enzymes in both the fed and fasted states is summarized here. In the fed state, when insulin is high, it's key to remember the acronym FED, dephosphorylated active. What is meant by that is that in the fed state, the enzymes that are supposed to be active are dephosphorylated to be active. Another way of saying this is insulin stimulates a phosphatase, which initiates dephosphorylation. This allows for the activation of PFK2, pyruvate kinase, glycogen synthase, pyruvate dehydrogenase, and acetyl-CoA carboxylase. These are the regulatory enzymes required for glycolysis, fatty acid synthesis, and glycogen synthesis. The dephosphorylation of phosphorylase kinase and glycogen phosphorylase inactivates these enzymes. In contrast, in the fasted state, when glucagon is high, glucagon activates a cascade that initiates the activation of protein kinase A. Kinase A will phosphorylate proteins and activate those proteins required for fasted state metabolism. It phosphorylates PFK2, which activates the phosphatase activity. The phosphatase activity is also termed the enzyme fructose 2,6-bisphosphatase. Remember, this is our bifunctional enzyme that's referred to as PFK2 or fructose 2,6-bisphosphatase. It also phosphorylates phosphorylase kinase and glycogen phosphorylase, which activates glycogenolysis. This activates the pathways of glycogenolysis, fatty acid oxidation, and gluconeogenesis. The other enzyme that's phosphorylated that's not listed here is hormone-sensitive lipase, and phosphorylation of hormone-sensitive lipase initiates lipolysis in the adipose. The phosphorylation of pyruvate kinase, glycogen synthase, pyruvate dehydrogenase, and acetyl-CoA carboxylase inactivates these enzymes. This ensures that the pathways active in the fed state are inactive in the fasted state. The last metabolic state we'll talk about is the starvation state. And this is a period of not eating that extends from days to weeks. Under these conditions, glucagon, cortisol, and epinephrine are elevated. Because of the extended period of fasting, glycogenolysis is not very active as the majority of glycogen stores have been depleted. You'll also notice that in comparison to the fasted state, gluconeogenesis is decreased to some extent. Red blood cells are the only tissue type that are still relying heavily on glucose as a substrate for energy. Lipolysis is occurring, which provides fuel for beta oxidation, which is occurring in both the muscle and the liver. In the liver, the products of beta oxidation are generating ketone bodies. These ketone bodies will be used as a substrate for energy by the brain. The transition of the brain to using ketone bodies as a fuel source, as opposed to glucose, allows for the sparing of skeletal muscle. In other words, as ketone bodies are used by the brain, the amount of glucose synthesized by gluconeogenesis can decrease to some extent. Therefore, the substrates for gluconeogenesis, or amino acids, can also decrease. So in the starvation state, muscle catabolism is reduced, which is consistent with a small reduction in gluconeogenesis. And this is as the brain transitions to utilizing ketone bodies as a substrate for energy. You'll notice that urea synthesis is also decreased, and this is consistent with a decrease in muscle catabolism. As fewer amino acids are released from the skeletal muscle to be used as a substrate for gluconeogenesis, there are fewer nitrogen-containing compounds providing substrate for the urea cycle. So under the starvation state, shown here, the nervous system, such as the brain, will be using glucose and, to a large extent, ketone bodies. This decreases the need for gluconeogenesis and reduces the level of amino acid catabolism. Other tissues, such as skeletal muscle and the liver, are primarily catabolizing fatty acids through beta oxidation, and again, the red blood cells will also be reliant on glucose. It's only under starvation states where ketone bodies will transition to be a primary fuel source for the brain. Within the liver, we know that the catecholamines, glucagon, and insulin can all elicit several different downstream signaling cascades that will impact the metabolic regulation of these integrated pathways. If we follow glucose in the fed state, we know that glucose can be taken up by the liver through the GLUT2 transporter. It's phosphorylated by glucokinase, and the transcription of glucokinase is increased in the presence of insulin. From here, glucose 6-phosphate can be used in the pentose pathway by glucose 6-phosphate dehydrogenase to generate NADPH and ribulose 5-phosphate. Alternatively, glucose 6-phosphate can be used to synthesize glycogen, and our regulatory enzyme here is glycogen synthase, which is active and dephosphorylated. Glucose 6-phosphate can carry on through the glycolytic pathway, and the first regulatory step is the conversion of fructose 6-phosphate to fructose 1,6-bis-phosphate. The enzyme here is phosphofructokinase 1, and its activity is stimulated by high levels of fructose 2,6-bis-phosphate and AMP. The conversion of PEP to pyruvate is regulated by pyruvate kinase. Pyruvate kinase is active when dephosphorylated. From here, pyruvate can be converted into lactate under anaerobic conditions, but the majority of pyruvate will enter into the mitochondria and be converted to acetyl-CoA by pyruvate dehydrogenase complex. This complex is also regulated by phosphorylation and by any number of allosteric inhibitors or activators. Acetyl-CoA continues on through the TCA cycle. The conversion of acetyl-CoA to citrate involves the condensation of oxaloacetate and acetyl-CoA. Citrate can continue on the TCA cycle or can be shuttled out of the mitochondria to be used as a substrate for fatty acid synthesis. Fatty acid synthesis in the cytosol of the cell is stimulated by high levels of citrate as well as insulin, where insulin will dephosphorylate acetyl-CoA carboxylase. Malonyl-CoA is used to synthesize palmitate, and palmitate is used in the synthesis of triacylglycerols, which are packaged in our VLDL particles and secreted from the liver as a means of circulating triacylglycerols for storage in the adipose. Alternatively, the acetyl-CoA in the cytosol, generated by this shuttling of citrate from the mitochondria to the cytosol, can be used as a substrate for cholesterol synthesis and the synthesis of bile acids, and we've looked extensively at the regulation of cholesterol synthesis. The only pathway that's not shown on here is the urea cycle, and the activity of the urea cycle, remember, is not hormonally regulated, but rather primarily regulated based on the amount of protein catabolism that's occurring. And protein can either come from the diet or from muscle catabolism. So the integration of these pathways, glycolysis, glycogen synthesis, mitochondrial TCA cycle, coupled to the electron transport chain, the shuttling of citrate, malate, or aspartate out of the mitochondria, and the cytosolic pathways of fatty acid synthesis and cholesterol synthesis should be very familiar now. To summarize this module, we're going to look at two different metabolic impairments that precipitate pathological symptoms. The first we're going to look at is the condition of diabetic ketoacidosis. And this is caused by a lack of insulin. So we'll first look at what happens when insulin is lacking within this system. The second pathway we'll look at is a defect in beta oxidation. So we will address what happens when the process of beta oxidation is impacted. And hopefully these two deficiencies will help you work with this material and become more familiar with the integration of the pathways.